or do anything?
Nope. You're good.
I'm good.
You're good. All right. Hi there, everyone. Thank you so much for attending Popping the Science Bubble. Thank you for bearing with as we just had no internet in the building for the past few minutes, and it looks like we're good now. So thank you so much. So welcome to Popping the Science Bubble. It's great to see an audience here. This is our second time doing this program in person. We've been doing it on Zoom throughout the pandemic, and we're really glad to have the program back. Popping the Science Bubble is a monthly seminar series that aims to share new research, findings from grad students and postdocs at UC Berkeley with the general public, and to create a constructive discussion about a variety of science topics. We also have people from Berkeley Lab who participate, and all kinds of Berkeley community members who contribute to this program. We have 2 speakers at each seminar who will talk about their current research or a topic that they find really interesting. The organizers are 4 graduate students at UC Berkeley: Helen, Daniel Bretthauer, Madison Lemur, and Oksana Andruk. Lectures happen on the 3rd Tuesday of the month at 5:30 p.m. Programs are usually live streamed on the Science Bubble YouTube page or Facebook, and you can view them later and share them with others. We're working on recording tonight's presentation, so it should be on the Science Bubble YouTube page to view later. You can also join the Science Bubble's listserv to be updated on new topics as they are being organized. So thank you so much. I'll let the Science Bubble team take it away.
Thank you all for being here. Again, thank you for holding on for us through technical difficulties. We're excited to be back. If you enjoyed tonight's talks, we're here every month, so we will put up our listserv and other socials at the end of the talk, so you can follow us continuingly. But for tonight's format, I just want to let you all know we're an open presentation format, so if you have questions during the talk, either a clarifying question or a question about a topic, please feel free. We will moderate so that you can engage as the talk is happening. But for our first talk tonight, we have Tom Elston. He's a neuroscientist at UC Berkeley. He grew up in Houston, Texas, and started his education at Baylor University, where he studied cognition-enhancing drugs. After undergrad, he then moved to Dunedin, New Zealand, where he completed a PhD in neuroscience at the University of Otago, studying the neural basis of economic decision-making. And then in 2018, received an Alexander von Humboldt Fellowship, which allowed him to spend 2.5 years at the University of, hopefully again I'm getting this right, Tübingen, okay, in Germany. So world traveled, where he is working on the neurobiology underlying decisions involving risks in both humans and non-human primates. He is currently now a postdoc in the Willis Lab here at UC Berkeley, where he focuses on the neural mechanisms of context-dependent choice in non-human primates. So we're excited to have him kick us off tonight. Thank you, Tom.
Thank you for that very kind introduction, and thank you all for being here. So, the main topic of my talk tonight is context in the brain, and before we really get started, I just want to give a very, very brief introduction into cognitive neuroscience. What is it? How do you think about it? And then how can we kind of use that to inform ultimately the research that I'm hoping to present to you? So if we just start with the question, what is cognitive neuroscience, we can just look at its 2 words. The word cognitive, essentially referring to hidden mental processes, like learning, memory, decision-making, attention, and so forth. And neuroscience, so we're interested in the physical workings of the brain. And whenever you might ask yourself, okay, that's all well and good, but how do we actually isolate some kind of cognitive process? How do we think about cognitive processes occurring in the brain? And a very influential framework for this is called the information processing model. Now, this isn't to say that there aren't other models, but this is the one that I personally like. And the basic idea is that we have the environment and we have the brain. The environment is everything that's outside of you, basically. And, of course, let's imagine that you see something in the world. So you have some sensation, light hitting the back of your eye, and you're kind of loading that information into your brain. Now then, once that information is actually in you, that's not enough to have intelligence and to respond. Now we need to have something that allows us to evaluate the world around us. And broadly speaking, that's the domain of cognition. It's that sweet spot of between sensing yet before responding. It's that thing that gives us a buffer so that we can intelligently respond to the world. And so to kind of illustrate just how, when I say low level and basic, what I really mean is we're doing this all the time, going through this cycle. Is imagine that you're at the grocery store and you are picking some cherries to eat later. And you happen to know that a sweeter cherry is a better cherry. So what's happening is that as you kind of look at it, imagine that my cursor is your fixation point as you look. Oh, you're like, oh, that one's pretty good. Less good, less good, less good. Oh, that one's really good. And so what's happened, each time that you moved your eye in that circumstance, you might imagine that you load in the environment, that immediate pixel level representation of just, I'm looking at that section of visual space right now. And then you evaluate that, right? How is that a dark cherry? Because you have some set of prior beliefs that are hanging out in here. And let's say that we're currently looking at this cherry. Well, that's a relatively light one, so that's not the one that we want to pick. So then we relay that over to action. We move our eye, and now the process begins again. And so the idea here is that this process of the information processing framework is essentially saying how it is that we're constantly loading in, evaluating, and responding to the world dynamically. And so when we design experiments to assess this sort of thing, what we really need to do is we need to design tasks. That's really what the core of cognitive neuroscience comes down to, is designing clever behavioral tasks which isolate a very specific aspect of the mind so that there's no ambiguity about what mental process the subject is engaged in when we make measurements of the brain. Now that we've had the incredibly quick crash course in what is cognition and so forth, now we need to think about how do we actually measure physically activity in the brain related to the mind. And so the brain is composed of billions of neurons, these incredibly tiny individual cells, individual electrically active units within the brain. And these units, as I said, are electrically active. So the idea is that imagine time zero is nothing's going on, and then what happens is that all these cells are hooked up to each other. So they start talking to each other and kind of accumulating activity. This is the activity of, let's say, just this neuron, which is getting input from a bunch of other ones. And then what happens is it gets 1, 2, 3 little bits of input. It hits some threshold, and that causes the neuron to what we call spike. So it emits this burst of electrical activity. And the key thing here is that spikes, the very, very fast electrical changes in these cells is kind of the currency. It's essentially the grammar or the language of the brain. And so one of the most popular methods to study the brain, which is not based on spikes, is called functional magnetic resonance imaging, fMRI. So this is the kind of image that you've probably seen in newspaper articles and so forth, where we have an image of a brain with essentially some activity kind of heat mapped over it. Now, the thing that you need to know when seeing this kind of an image is that this heat map doesn't actually reflect the activity of neurons. What this is measuring is the flow of blood in the brain. And the idea is that it's an indirect marker of brain activity, because whenever a neuron becomes active, let's say here's the non-active case, so nothing's going on, but whenever they become active, the neurons require oxygen. And what this signal is really picking up is the increased blood flow and therefore increased oxygen moving into the brain. But there's, in my view, a relatively major issue with trying to study the brain with this non-invasive MRI technique. And that is simply that it's really, really slow. So here on the y-axis, we have blood flow, so just how quickly it takes for blood to kind of move from one spot to another to indicate neurons are active. And you'll see here that the units of time on the x-axis are now in seconds, whereas spikes are occurring on the order of milliseconds. So the fact is that, if we think back to the example of picking cherries, you might move your eye 10 times within 5 seconds. So the fact is that this kind of a signal with MRI is just going to completely miss the boat with actually capturing the unfolding of a mental process. Yeah?
Is that time exact, or is that just like to show the order of magnitude?
It's to show the order of magnitude, but it's about that ballpark.
Okay. But we don't know exactly how long it would take from like an event happening exactly to the blood flowing to the place that would be responding to it.
I mean, we can definitely measure that, and for individual instances, we can know that. But of course, there's going to be a little bit of variability from moment to moment as this happens. So I don't think giving you, let's say, like an exact number is all that useful. It's much more helpful to think about it like, here's some average number with a bit of error around that. And this around 5-6 seconds is pretty typical for such a delay, essentially. But what this really gets at, though, is that what we need to do to really understand the physiology of the mind, to understand how it is that the brain implements the mind, how it is that cognition unfolds dynamically as we interact with the world, we need to directly study neurons. There's no way to observe this kind of thing, and therefore to address the diseases associated with cognitive decline and dysfunction through something like fMRI. We must measure the neurons. And so, I kind of just want to give you guys this context because this is really the driving force behind why I use non-human primates in my research, is because they have the intelligence at a similar level to humans in many different domains, but we're able to directly measure their neural activity. And so we're kind of able to get the best of both worlds. So now I want to tell you about my experiments about how context is represented in the brain. So imagine that you have just decided to go on a holiday, you have some extra money to spend, and you are going to rent a Corvette. So lucky you. And the biggest challenge, the biggest choice facing you in your life right now is, do I get the convertible or do I get the hardtop? A problem we all wish we had. Now, normally if someone were coming to the Bay Area, it doesn't rain that much, so in which case the convertible is probably the way to go. But on the other hand, if you are making a road trip around New Zealand's South Island, and you're going to go to New Zealand's west coast, it rains all the time. And therefore getting the hardtop is probably the way to go. So the idea here is that the best choice depends on the context. As you're kind of evaluating your choice options, you need to contextualize them to figure out what's good and bad. And this is really the guiding question for my work for this project, is how does the brain allow context to, how does this happen in the brain? So when we think about where to look, how do we begin addressing this question? There are really two brain areas that would first come to mind. The first one is a brain area called the hippocampus. And it's particularly well known for its dysfunction in Alzheimer's disease, and its atrophy is strongly associated with the onset of dementia. Now, the reason we think it's involved in context is because it contains neurons that are called place cells. And I'm just going to illustrate here two examples of these cells. So imagine that this is one neuron, and it's recorded from the hippocampus of a rat as it walks around a circular tank. And every little pixel in here is telling you how much the neuron fires when the animal is in that spot. So what you can see is that when the animal's in the circular environment, cell 1 and cell 2 both fire quite a lot when the animal is in this top bit of the environment. So you can actually decode where the animal is. A lot of people have thought this might be like the brain's internal GPS system. But what's kind of wild is that if you take the same animal, same neuron that you're monitoring, and now you put him into a triangular environment, this cell basically transfers its activity. It's not firing at the top of the environment anymore. Cell 1 starts firing at the lower right, cell 2 starts firing, somewhere up here in kind of upper left. And the idea here is that if you take the animal and go back to the circle again, this just snaps right back on. It's as if the brain carries these different maps of the environment, and it keeps track based on which environment that you're in. That kind of makes sense? That when you're in this context, the cells represent the environment this way. When you're in this one, they completely change the way that they encode the space, and you can go back and forth as many times as you want. Now, when we think about decision making, another major area people think about is called the orbital frontal cortex. And that is basically the bit of the frontal lobe right behind your eyes. Hence orbital, eyes, frontal, front. And here just what I'm showing you is that if you inject some radioactive tracers into the hippocampus and see just where those neurons project to, you see that they are densely projecting to the OFC. That this is the bottom of the brain. It's just kind of turned upside down. You can see these black bits are indicating that that's where the neurons are projecting. So these two areas are really hooked up. And what's kind of wild is that patients who have damage to this brain area just make terrible life decisions. So there's some really entertaining stories and sad stories of this patient, EVR, who was an actuary in a town. And I mean, it's a pretty brainy job. He then had some serious brain damage to his OFC. And all of a sudden, he just started making terrible life choices. So he all of a sudden spent all the town's money on the wrong stuff and almost made the town bankrupt. He divorced his wife of 20 years and married a prostitute. He then got fired from his job as an actuary and took a job 100 miles away and then got fired for being late all the time. And so it's just critical that something about the OFC allows us to make good choices. Now, down the track, people have been able to actually record the activity of individual neurons in the OFC to kind of understand how is it that the OFC allows us to make good choices? Why is it important? Mm-hmm.
Do we know if this area in the brain is what fires if you see new information as well? Or if it just doesn't activate if it's like a whole new scenario?
So, short answer is no. I should say we know the answer, and that's not what it is. But it does seem that the brain places what you could kind of call like an information bonus on new things. And I think let me explain this bit and then kind of tell you what that would mean if we could see that on this graph. So imagine a scenario here. This is the activity of one OFC neuron recorded from an awake behaving monkey making decisions. And the idea is that he can pick different choice options that pay off different amounts of juice. And monkeys love apple juice. They have it almost is like a little beer hat, and they're just getting different drops, different numbers of drops of juice squirted into their mouth, and they are all about this. And what you can see here is that if the monkey picks something that's going to give him 8 drops of juice, this neuron just drops off its firing. And if you see, quite a lot. And if you pick something that's worth 4 drops of juice, it drops less. If you pick something that's worth 2 drops of juice, even less. And the idea is that based on just the activity of this one neuron, you can actually read out the value that the animal assigns to the different choice options that he might select.
Mm-hmm. Is the y-axis here
oh yeah, I should.
Current?
No, the y. I didn't.
It's signal response.
It's the firing rate. So
okay.
Earlier, I didn't realize that I had cut that off. But earlier, when I was showing you that spike diagram, you can imagine this is the number of spikes per second, basically the discharge rate of this individual cell. And so here, what we're seeing is that subjective value is encoded by the firing rates of these individual neurons. Now, with the idea of the information bonus, what you can imagine is that you can take a value function like this, and you basically just turn it up. So information just kind of blows it out even further. It accentuates an already existing value motif in the OFC. So all of this kind of sets up for me the question of, is contextual information influencing value information? And is that a way that we're able to use context to reason whenever we need to make value-based choices? So to address this question, I trained two non-human rhesus monkeys, to perform a context-dependent valuation task. So the idea is that the monkey is sitting in a special chair. He's looking at a computer screen, and he has his little beer hat on to get his juice when he makes good choices. And I'm tracking his eyes. And he's making his choices by staring at things. And he's learned how to do this. The way it works is that he begins by fixating on a little point of light. And then I show him a symbol that indicates, hey, we're in situation A or we're in situation B. And you'll see that there are two symbols for each context. And this will be important later. Then there's a brief delay. And then he's shown two emojis, postbox and lollipop, but you'll see there are 8 different ones. And these numbers here indicate the number of juice pulses he gets if he picks it. So the idea is that if you're in context A, and these are your two options, postbox paying 1, lollipop is paying 4, so clearly lollipop is the best choice if you were queued on this trial to be in context A. And if we're in context B, it's the exact opposite. It's saying that now postbox pays 4 and lollipop pays 1. So back to this idea of what does it take to make a cognitive neuroscience experiment, there's no other way to make good choices in this task. The correct answer is entirely determined by this context cue. So the only way the animal can solve this task is that first you must load context information in and use that to update the way that he's going to assign value to these choice options. So here what I'm just showing you is an example of one of my monkeys, Don, who can do this really, 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 really well. What we have on the x-axis is just the difference in value between two choice options. So, for instance, if the value of the left one is a lot worse, so negative relative to the right one, he basically doesn't pick it. And if the left one is better, he basically always picks it. So this is saying that this monkey really understands this task, and he's able to use context to drive his choices, which now means that we can start thinking about how can we monitor neurons within his hippocampus and within his orbitofrontal cortex as he does it. So now just what I want to show you is what's the process of actually getting this data? How do we actually measure neurons in the brain? So the first thing that we do is we get an MRI of the monkey, and we identify the different brain regions of interest. So here, this little bit, I've just segmented the OFC, so you can see it's at the bottom of the front of the brain. After that, I use that to design a chamber, which will then be implanted physically onto his head, and then I will remove a section of the skull, which will allow me to drive electrodes directly down in. Then I import the MRI into a computer drawing software, and then I design and 3D print these recording grids. And the idea here is that this recording grid, imagine it looks like a little silo that has a screw in it, and on that screw is attached something called a shuttle. It's a little plastic bit that can go up and down. And then we glue an electrode onto that shuttle. So that means by turning the screw, the electrode goes up and down. Does that kind of make sense? And by doing this with 3D printing, this gives us an extremely high degree of precision to target almost anywhere we want within the brain, while using only a relatively small chamber to do so. In the past, we would have needed much bigger chambers to record from these different areas.
Are the regions of the brain for a monkey the same as human?
That's a great question. Yes. And that's really one of the major selling points of using the non-human primate model, is that they contain brain areas that we have that rodents don't. The connectivity between the brains, the different brain areas of primates, is incredibly similar to that of humans in a way that with rodents it's just not the same. So
like 99% similar?
It's super duper similar. I don't want to give you an exact number, but it is overwhelmingly similar. And I think what's I should really also emphasize is that work from non-human primates has led to the development of motor prosthetics that's allowed people to control robot arms with their mind. And it's led to the development of deep brain stimulators that has cured the tremor associated with Parkinson's disease. And none of that would have been possible if we had used a different animal model to study the brain. But of course, before we can make these cool devices, we need to know how it is that these different parts of the brain work. That way we can then later design a device to interact with the brain. And that's what these recordings really allow us to do. And this is just what the electrodes look like. So it's essentially a little stainless steel needle that has 64 microelectrodes printed down the bottom of it. And this allows me to monitor 100 or more neurons at a time in different brain areas. Now I just wanted to show you, this is an example of 2 neurons on 1 electrode. So what I'm about to play for you is a video of a real neuron that I recorded from this animal. So this is an oscilloscope that's just directly patched into the electrode. And what you're actually going to hear are these little popping sounds, and those are the spikes. And what you can see is that there's a big neuron right here. You see the spike right there? But there's actually a second smaller one that's kind of going up in a slightly different bit. Every neuron has, you could almost kind of imagine, like its own fingerprint, and that fingerprint is its waveform of activity. So here's what it sounds like. I'll play it again. So in a bit, what I'm going to be going into is what are these neurons doing? Because the trick here is that information is actually represented at that level of that super high intense, and it's just modulating that firing rate up and down. So the first thing I want to show you is how it is that context is represented. So in my task, right, I've got these 2 different cues that mean context A and 2 different cues that mean context B. So that should mean that if a neuron is encoding context, it's going to fire the same way in response to either of these stimuli, right? The idea is that it doesn't care about what the stimulus looks like, it cares about the concept of context. And here's an example of a neuron in the hippocampus that actually cares about this, that does this. So you see, this is time on the x-axis, firing rate on the y. You can see right as the context cue turns on, this neuron preferentially increases its firing for both cues for context A, and doesn't really increase its activity for context B, but the idea is that this neuron, you can actually decode what situation the monkey is in on this single trial from looking at this neuron. So it kind of makes sense that this is like the building blocks of how information is represented in the brain.
Hang on, waking and sleeping, what's the, when you're awake or asleep?
Like,
Is it on and off, the neurons, when you're asleep or awake?
that's right. So, I mean, you'll see that it's not like the neuron's firing rate is ever at 0, right? Even its kind of baseline, the lowest it ever goes is like 2 spikes per second. So it's not to say that the neurons are ever off. It's just to say that when you use a particular mental function, it brings online a particular set of cells. And those cells, in many cases, are active in the same way when you're sleeping as when you are awake. And it's widely believed that that similar replay of activity patterns during sleep is the basis of dreams. But now just what I want to indicate here is that in the orbital frontal cortex, if we look at this cue period, it doesn't do that. The orbital frontal cortex cares about what individual stimuli look like. So what this is already telling us is that neurons in the hippocampus initially encode context. They're the guys that basically load in context information into the mind. Now what you see though is as the choice options come online, that's when the orbital frontal neurons start doing this. So it's basically saying that the hippocampus extracts context information from the environment. And it's like, hey, I realize I'm in context A. And then when you need to use that information, it's represented in the PFC, in the same place where these value encoding neurons are stored. And this is just a summary, basically, just to say that about 20% of the neurons in hippocampus encode context at the time of the cue, and about 25% of neurons in the OFC encode context when it's time to use context to make a choice. And this is what we call a double dissociation, basically saying that something happens in one brain area when you're using one type of mental function, and information is represented in a different brain area when you're using a different one, loading context in versus using context to make a choice. Now just what I want to show you is what are the consequences of this, because I told you in my talk advertisement that I would talk about contextual reasoning, and what does that really mean? So now we're just going to focus here on OFC neurons, when we know they're maximally active, the time when they're maximally representing context when you're deciding. Now we're going to look at how it is they represent value within each of these contexts. And remember, value corresponds to the number of juice pulses that the animal gets for making a choice. So this is one neuron. We have time and activity rate for the neuron. And look at this. When the animal picks a stimulus worth 1 pulse of juice, it fires very little. When it picks something that's worth 2, it fires more. When it picks something worth 3, it fires even more. And when it picks something that's worth 4, it fires even more. So you can actually linearly read out the value of the choice the animal is going to pick. Yep.
So this is still a neuron firing rate. Where is it in the brain?
This is the orbital frontal cortex. So this is basically, imagine this gray line is like the average of all the neurons. This is kind of giving you one example of one of those neurons. But yeah, in the prefrontal cortex. So it's at the bottom of the front. Now what's kind of wild is that if we look at the same neuron in context B, we still see that it's activated at the time of choice, but it's all out of whack now. Because look at this. Now 4 is at the bottom, then 2, then 1, then 3. So there's no meaningful value code here. What this is basically telling us is that this neuron only encodes value in context A. And then we have another example here of a neuron that only encodes, so this is a different neuron also in the prefrontal cortex that only encodes value in context B. Look, 1, 2, 3, 4 in context B. But in context A, it's 4, 3, and then doesn't differentiate between 1 and 2. Mm-hmm.
So did you
Go ahead.
Did you just have to move around the probe until you found the meaningful ones?
No. The idea is that we're trying to record a whole bunch at the same time, and whenever we are putting the probe in, we're not making any kind of judgment about what the neurons are doing before we hit record. All we want to do is just randomly sample what do the individual neurons in this brain area do. And then after the fact, we statistically assess what do they do, and that's how we infer this is the function of this brain area.
How do you distinguish one neuron from another in that context?
It's a great question. So if we go back to this, you know how they say every snowflake is unique?
Mm-hmm.
You can also kind of think about every neuron's waveform is unique. So if you look really hard here, there are actually 2 neurons that are being represented that you can see in this scope. So we see both of them. We have the initial shoulder, so that's the integration, right? Something caused it to fire. Then it spikes. There we go. Then it spikes, so we see that. And then one neuron comes up really far, but we can see there's another one right here. You see that? Yeah, it's kind of hard to see it, I guess, because you can only the screen when I was recording, it refreshes every 2 seconds of the activity. But when you look at the activity over like a 5-second window, or I'm sorry, bigger time window, it becomes much more distinct. And we have a whole process of something called spike sorting, which is where we identify templates of each neuron's kind of fingerprint, its waveform shape, and then we assign, because you could think here, every individual little line here, every thin little white line, that is one event, one spike for one cell. And so after the fact, it's a fairly painstaking process to go and assign each of these lines to say, hey, this spike came from neuron 1, this spike came from neuron 583. But that is a labor of love that we call spike sorting. Yeah, go ahead.
Do you have to factor in the left-right hemisphere stuff?
So we do, and that's also kind of cooked into the design here. Let me back up. There are differences between the hemispheres, mainly for visual processing.
So for your purposes, pretty much doesn't matter.
That's right, and we counterbalance it. So the idea here is that the context cues, when only one thing at a time appears on the screen, it's in the center, so it gets equal access to both. And for the choice options, we always make sure that they can be presented on either side of the screen. So there's no way that, for instance, good things always appear on the left, or lollipop always appears on the right. It's completely intermixed so that it's going to average out when we have hundreds of trials, because the animal's going to make maybe 1,000 choices in one sitting.
Question.
Yeah, please.
Does it make a difference before the experiment and after the experiment? In fact, like the response change, or the monkey kind of learn how to associate the context?
Yeah, so I mean, they definitely don't. They're not born knowing how to do this. That's for sure. And it is. Anyone in the monkey business will tell you it is a major effort to train them to do this, because they have no idea what a chair is. So you have to even start from that level of, this is what a chair is and this is how you sit in it. But so it took me probably 4 months to get an animal to do this at this level of accuracy. But once they have it, once they know how to do it, they can probably do it for the rest of his life.
So before that, these 4 months, when you're looking at these neurons, they are just random. They have no association to the context.
Well, that's one of the other big things, is that we definitely are not going to put the electrodes into the animal's brain without a very good reason, right? So we wouldn't, essentially that's not something we would measure, because imagine this. Imagine that if we just want to put the electrodes in, go through all the surgical procedures and all of that to measure neural activity without a hypothesis, without something clear to link it to, that would be cruel, right? It would be an unnecessary burden for the animal. And so for us, it's very, very important that we only go through all of this when we have overwhelming evidence that the animal knows what he's doing, and that we're going to be able to measure something that previously would have been impossible or that had never been seen before. That's the only reason, the only justification to put the electrodes in and to go through all of this.
So that's why you showed us this graph first, is because that's like how you know that the experiment is valid, and then you're measuring it.
Exactly. We know that because the animal is always picking, well, he's never picking the left thing when it's bad, and he's always picking the left thing when it's good. That means that we know unambiguously, without a doubt, he's using context to influence his sense of value when making choices. And if we didn't know this, there's no way we would proceed. You can kind of imagine that most neuroscience experiments die at this stage, because you just realize, wow, the monkey is not doing what I thought he was doing. It's only whenever it's unambiguous. This is the mental process that I've been able to isolate with this task. So here I just want to kind of have an intermediate summary of just what I've shown you with the single neurons. This double dissociation of hippocampus encoding context and OFC using it, and when it uses it, it's bringing online different subcircuits for value, different non-overlapping ensembles of neurons. But the big question here is, are these two regions of the brain communicating? Because it does kind of seem that way. We know that they're connected. We see it start in one spot and then move to another spot. So to assess this, we measure brain waves, what are called oscillations in the brain. And this is a very famous video just demonstrating how it is that oscillations represent the synchronization of cells, so they fire all together. So in this little video, you're going to see a very famous neuroscientist named Robert Desimone. And he has a bunch of metronomes. So imagine each of these metronomes is like a neuron. Every click is like the neuron firing. And at first it's just going to seem totally random. And then when he physically joins them, as they in fact are in the brain, you'll see that they synchronize. And this is what a brainwave is.
Our oscillating neural network demonstration. Let's get these oscillating. Okay, they're actually random with respect to each other. But now let's give them a weak physical coupling on these drink cans.
It's crazy, right?
There they go.
Pretty well.
They're all oscillating in synchrony. That guy on the end.
What does the cans?
Yeah, what does the cans have to do with it?
Close.
Well, it basically allows the activity to slightly transfer. It basically allows the motion from the metronome to go from the metronome to the plank and then onto the cans. And because the cans can slightly move, it's like summing or averaging all the motion. And therefore it kind of buffers and allows them to synchronize. So it's crazy, right? But the idea here is that when you think about what is a brainwave, brainwaves emerge from the synchronized activity of neurons. And so what this kind of looks like,
We're going to try our oscillating neural network next.
it's cool, right? And so here, basically what I've just overlaid is actual brainwaves that I measured as my monkey performed the task. And so what you can see is that it really does just look like an oscillator. It just really looks like a sine wave that's going up and down. So this isn't the activity of one neuron anymore. Now we're talking about the summed activity of many neurons all together. And the idea here is that if we see two brain areas that kind of oscillate or they kind of hum together, that's implying that they're sharing information. They're somehow coupled and information is moving from one spot to another spot. So now just what I want to show you is what this actually looks like. So here on the x-axis we have time, where 0 is when the pictures turn on. The y-axis here are individual trials, so individual bits where the animal is going through this trial structure. And the color is basically the phase, the different part of that brainwave that's going on. And if you kind of look at it, you can almost imagine it's just, this brain activity is just rolling on through. See what I mean? But it's kind of inescapable to see this bit here, what looks like this huge, almost on every trial, this big wave crest that basically happens. Imagine like you're going surfing. Yeah.
So this is this, like we should compare this like in the times to the one we saw earlier. Like so where that thing spiked, it's right there.
That's right.
That's
Basically, as soon as the context information is coming on, where you have 20% of hippocampal neurons carrying this information, it induces this huge wave of electrical activity across the hippocampus. And what's really interesting is that it's also showing up in the orbital frontal cortex, also right as that context cue is turning on. And so here this alone is telling us that there is likely some kind of synchronization between these structures. And this is what makes you think, okay, now I'm going to try another kind of analysis called coherence analysis to actually quantify, are they in fact oscillating together? Are they communicating? So this kind of a graph is called a coherogram. The idea is we have time, the different frequencies, so how squished together those oscillations are. And the color is telling you how coherent they are. Are they going up and down together? How well do they go up and down together at a point in time? And what you basically see is they start becoming very coherent right as context Q turns on. It ramps up and basically peaks right as the animal makes his choice, right as those two different value subcircuits in the OFC come online. And so, through some fancy math, I'm able to model the direction of the synchronization. Who's leading? Who's following? And I won't belabor you with all of this, but the idea is that we can see that it's going from the hippocampus to the OFC. It's suggesting that something is being sent from the hippocampus to the OFC. And granted, because we're able to monitor the neurons, it's context. It's got to be context. So, to summarize, what I've shown you is that the hippocampus initially encodes context. Yeah.
So the reason why we're looking at phase is because that would mean that the things are happening at the same time.
That's right.
And not just because they're always oscillating, so phase would be the synchronization.
Exactly. It's saying that these regions are becoming temporally synchronized on the order of 100, less than 50 milliseconds of difference between them. They're super tight.
Mm-hmm.
And what's kind of cool then is that you can imagine down the line, now, okay, we know that this is a marker for how brain areas talk to each other. And in this particular circumstance, we know what's being sent. So down the track, what I would like to do is to develop a brain-computer interface that will allow me to read out in real time the communication between brain areas and then use that to basically enhance or to disrupt activity in both. So basically turning up or turning down the communication between these structures. And when you think about why this might be really useful, is that you think about people who have autism, for example. They often have the experience of sensory overload. The world is just too intense. They're unable to filter the sensory world. Too much is getting through. Well, maybe if we understood better how it is that a prefrontal area, which is getting the information in the end, communicates with a sensory area that's initially loading in the world, maybe we could actually selectively turn up or down how information is actually sent through. And that then might allow us to actually reduce the experience of sensory overload in people with autism. And so with that, I just want to thank my mentor, Joni Wallace, our two excellent graduate students in the lab, Nate Burnett and Eric Koo, and of course, our funder, the National Institute of Mental Health. With that, thank you very much.
I think we have a few minutes for a couple questions, and then we'll move on to the next talk. And I think Tom will stick around, so if there are any questions after that, we can also ask at the end.
Mm-hmm.
Yeah, sorry, you can start first.
Yeah, anything you want.
That was a really cool talk, first and foremost. In some of the plots that you had for, say, I think it was the flux of signals, or I guess any of these would probably work.
This one, for example?
It was sort of in the earlier slides, but any of these really would work. At the beginning of your talk, you mentioned that with MRI, part of the
Yes.
limitation of the technique is that the range in which you get a signal, at least on the x-axis or time-wise,
Yes.
it's in the seconds range as opposed to milliseconds range, or millisecond range.
Yes.
Do you think it seems like the signals, or the, let's say, full width at half max of these signals that you have presented on are sort of in, maybe not the same, but in the same order of magnitude of width. Do you think that there's any information that might be getting lost with these electro systems?
No, I mean, you could kind of imagine that as far as neuroscience techniques go, there's electrophysiology, monitoring ensembles of individual neurons at a time electrically is, you could kind of imagine as like the Lamborghini of neuroscience techniques. There's another very popular or growing and popular method called calcium imaging, which is where, when this spike happens, there's a bunch of calcium that enters the cell, and people have made genetically fancy mice where their neurons turn purple as that happens, and you can implant a microscope in their brain and make videos of neurons turning purple. And that's also, we could kind of say that's, if electrophysiology is the Lamborghini, then we could say calcium imaging is kind of like the Ferrari. The downside with calcium imaging is that you can't go deep into the brain. You can only really measure stuff on the surface. So you wouldn't be able to do this experiment with calcium imaging. Another kind of major issue I didn't highlight with MRI is that not only do we have a lot of temporal smearing, where you actually can't see things unfold really, but there's also a lot of spatial smearing. You think one neuron here, one teeny tiny little neuron, there might be 20,000 of them inside of one voxel, one pixel of this activity map in the MRI. And you think that each neuron does something a little bit different. So it's, in my view, and I think for many people in electrophysiology, it's a bit controversial about what this signal really means. And I think also, like when you might see an article like in the newspaper or something based on fMRI, you might think twice about the limitations of the technique before completely accepting it.
Thank you.
I think I'm a little confused about MRI and when they put a lot of little electrodes.
Right, so that's called EEG. And EEG fundamentally is representing...
We're going to try our oscillating neural network demonstration.
These waves. And the idea is that for some neural events, the signal is so big. Imagine there are so many metronomes. The Coke cans are so good that enough of them synchronize to make an electrical event detectable through the skull.
With the little electrodes.
Yeah, with the
but
little...
It's not three-dimensional. It's not telling you where it is in the middle.
It's not telling you where it is, and it's the average of a whole lot of stuff.
Okay.
So for some things, EEG is very useful, but its major limitation is that we can't know really where the signal is coming from.
Got it.
But it's non-invasive.
But it's non-invasive. And that's really the trick here, is that these kinds of experiments that I do are invasive, and I should say that it's a privilege to do it, because there are other techniques like MRI, like EEG, but the fact is that in order to justify the use of an animal and to do this kind of an experiment, we need to be going after a question that would just be entirely impossible to address in any other way. Oh yeah, like our animals. Well, for one, there's no nociception in the brain, so when the electrodes go in, it's not physically possible for them to feel it. So they don't experience pain. And then at the end of the experiment, at the end of their research time in the lab, we send them to a sanctuary. So we do not harm the animals, and we don't kill them.
Well, I'm wondering why funding from the NIMH as opposed to, I'm thinking this has potential for, say, stroke victims, survivors.
Oh yeah.
So why did you use the funder or apply to the funder in a mental health track as opposed to physical health track?
In large part because we're trying to focus on the basic mechanisms of cognition. So, most of my work, like for this context-dependent choice task, I kind of sold it, if you will, with respect to schizophrenia, because contextually inappropriate behavior is one of the big hallmarks of schizophrenia. So for instance, I witnessed someone about a month ago having like a full-on discussion with a wall in a parking lot. And in that moment, to me, it was fairly obvious that this was an out-of-context, this was a contextually inappropriate behavior. So somehow or another, their ability to use context clues to understand what's normal and what's not, and how to guide their behavior was entirely disturbed. But yeah.
Okay, I think just for the sake of time, we'll end this question period there. But please join me in thanking Tom so much for a wonderful talk. We'll also hold an open period if there are lingering questions afterward. But for now, we're excited for our second speaker, and then I think we'll go
Okay, well, Alexia sets up. I am going to, first of all, thank Tom for a wonderful talk, and I'm going to introduce our second speaker, Alexia. Alexia is a chemist with primary research interests in the field of metals in medicine and the ability to fight cancer using chemical design. She received her BS in chemistry from the University of Oregon prior to earning her PhD in chemistry from Stony Brook University in New York, where her dissertation investigated using radioactive light to eliminate cancer. Dr. Cosby is currently a postdoctoral researcher at Berkeley National Lab, where she continues to explore the interface of chemistry and nuclear medicine. She's excited to break down the stigma around radiation and share interesting facts and tidbits from her work at Berkeley Lab. So welcome, Alexia.
thanking audience. Also have USB-C if we need to connect it some other way.
Let's try that maybe.
Yeah. Sorry, I don't know if this is turning down the volume. Progress. There we go. Okay. Gonna turn so I can see everybody. Hi everybody, I am Alexia. Thank you organizers for having me. I'm excited to share a little bit about my work. I really thank you, Tom, for that talk. That was really great. Those brain images, the MRI images, maybe have that in your mind a little bit about what I'm going to talk about. So my work deals with radiation, but I am formerly a chemist by training. So a lot of what I'm going to be showing you today is some of the things that I find the most interesting about what I work with in the context of being a chemist. So kind of my talk overview to give you some, to contextualize this, I'm going to start from like talking about radioactivity fundamentals, just the basics, and all of this this overview goes into the design of my research and what I'm interested in. So you go from the fundamentals of radioactivity into chemistry design, and then how do those things interact together to discuss preclinical medical research, and then that leads eventually, hopefully, to clinical or FDA approval of your drug. And then ultimately what we're trying to spread with this type of research is global treatment and awareness of the current diseases and cancer research that's going on. So when we think about radioactivity fundamentals, you probably have some kind of image like this, right? Suited up, like you have images of maybe Chernobyl. It's like scary, crazy. How do we handle it safely? And I'm here to say that from a preclinical and chemical standpoint, there's a lot of safety controls that we have in place when we do this research. So this is not, maybe what you think of initially is not reality in practice of when we're actually going in and trying to do this preclinical research. So when you think about radioactivity, you need to be thinking about what type of radioactivity you're working with and how to shield yourself. So just on a very basic level, there's three primary types of radioactivity. As a radionuclide or as an element is unstable and it's decaying, it'll give off either an alpha particle, a beta particle, or a gamma particle.
And either paper, plastic, or lead, concrete, depending on what you're working with, these are things that can shield these different types of radiation. So up at the lab, what we use are these survey meters, and so I almost brought a lab coat today. We put on a lab coat, we put on one layer of gloves, we put on tape, and we put on a secondary pair of gloves. Underneath our gloves, we wear finger rings to monitor the dose that our hands are receiving, and then we put a little clip-on monitor on our lab coat. And so right after we're finished with our work, or if we're concerned we might have contaminated ourselves, we simply will take our outer layer of gloves off, and we'll take the survey meter, and we'll survey ourselves. And we're not throwing activity on ourselves. This is just the preliminary precaution of what we do. So in the context of medical applications, we want to think about, sure, we have alphas and betas and all kinds of cool things that these isotopes are giving off, but we want to think about specific properties of these isotopes and how can we actually use this, how can we contextualize this radioactivity into something that we can use. So there's two types of beta, and one of the beta, positron, can be used for imaging applications, and the others can be used for therapy. So I'm just emphasizing here, awareness is key, contextualization is key, right? The type of radiation you're using, the half-life. Some radioactive elements decay immediately. They have a really, really short half-life. Some live for millions and millions of years. And one of the really, really cool things about the research that I do at Berkeley Lab is I'm uniquely posed to be able to work with both types. Not all these labs and all of these spaces have the facilities or the ability to work with a really short-lived radioactive nuclide and a really long one. So with that, the other things that you want to keep in mind when you're working with radioactivity is just ALARA, as low as reasonably achievable. Give yourself time, give yourself distance, and give yourself shielding. So those are really just the basics about when you're thinking about working with radiochemistry, and I wanted to show you kind of contextually what this work looks like. It's not anything so crazy as to like suits and hot cells, and I mean some maybe, but some of the work I've done, here's an example of a fume hood where we do chemistry, and when we do radiochemistry, for example with our positron emitters, the only way you can shield them is with lead. So what I'm showing you here are these lead bricks that we line up, and then inside of the lead is where we do our chemistry. And that way, you're completely shielded by any of the damage that beta particle is emitting. So a few other things, right? Lead shielding. Dose calibrator, once we are done doing our chemistry, we can take a vial and simply drop it into the dose calibrator, and it will tell us how much activity there is. We also have, plastic shield. We're working with radioactivity, but we're also working with chemicals, so you don't want to be splashing the actual chemical hazards on you. And then other, radiochemistry or pipetter type tools. So this is basically a summary of if I'm ready to go start doing radiochemistry, this is what my hood looks like. So I want to talk about radioligand basics. I'm going to give you an example of a type of radiochemistry that you can do to try to image and treat different types of cancers in a very general sense. So this is done basically by chemical design. You take a targeting vector, and this is a well-known thing that we know is going to go find the cancer. We attach it via chemistry to a linker, and then this gets tethered to our radioisotope. And so this whole construct right here is what I will show you examples of. There are FDA-approved examples of these constructs that are put into people that contain this exact design. So there's a lot of different, we can change it out. There's a lot of different systems that we can use with this. There's a different types of targeting vectors, depending on what kind of cancer you're going after. This right here is a general symbol of an antibody. Antibodies are,
what scale is that? Because that looks like a protein, but so is it what scale is it on? Is it atomic? Is this 3 atoms, or is it larger like a protein?
Yeah, so that's a great question. Contextualizing this is maybe a small protein. Small proteins are going to travel through your body more quickly, so it kind of depends on what sort of cancer you're looking at. This here is an antibody, so many more proteins. They're not scaled at all. Antibodies are very large, and they travel through your body much more slowly. So you kind of need to pick your targeting vector depending on what type of cancer you're going after. A lot of the most common models that we work with are prostate cancer and breast cancer, and that's just because of their prevalence. So then you want to pick your targeting vector, you want to design your construct, and then you want to think about what kind of experiment am I doing, what kind of question am I trying to ask? Am I trying to image cancer, or am I trying to treat it? So this, not to get too chemistry crazy, this is what a compound like that is FDA approved looks like. This right here is the targeting vector. You can simply buy this thing as a chemist if you're trying to make it. You basically can just buy it. You then do a little bit of chemistry. It's like one reaction. You step into your hood, mix them together, then it's done. Probably takes 24 hours. Linker can be made or purchased. And then to finish assembling this construct to inject into a human or inject into a mouse, you have this chelator. A chelator is like a little basket or like a baseball mitt. If the baseball is your radionuclide that you want to have firmly attached, then the chelator is the mitt that just holds onto it. And so you make the chelator, you assemble this whole crazy organic molecule, and then you throw the radio isotope at it, and it's stable, and it's all pieced together. And so this is a lot of chemistry mostly, but it's also biology, thinking about what kind of targeting vector are we using. So this leads into this really interesting form of research called theranostics, and it's using both cancer diagnosis and therapy in one treatment, in one molecule. So the idea is to have first diagnose cancer by imaging, follow up with therapy, and then follow up again with imaging. And I will show you an example of that in a second, but this is, the idea behind this is to streamline personalized medicine so that when you go and you have, you go and get a checkup, you can be given the same thing each time. You don't need to go for multiple different types of treatment. It's personalized to you. So I'm going to take this exact FDA approved example here. So here is a patient who received imaging with this gallium dotatate, and gallium-68 is a positron emitter, so you can image with it. And so gallium-68 just gets thrown in here, gets recognized by the molecule, and then you image. Yeah.
So the radioactive molecule is unstable, and so you send your linker, it goes to the right place, and you have to figure out how long it'll take, and that's part of the experimental design, and then it decays, and that's what we're measuring?
Yes. So exactly. So this patient here has all of these different tumors, and the tumors express something. It's like, negative signal that this warhead thing, and that's biology, you don't understand that quite as much. It's saying, hey, let's recognize it. They literally attach their receptors like this. And then the signal, what we're measuring is the positron emitter right there. That's what gets picked up in the scan. So depending on what kind of decay mode you're using from your radionuclide, that's the one you're going to put in. So gallium-68 is a beta emitter. We use it to image. Here's all this cancer. Then we use lutetium dotatate. All we're doing, notice my cute little animation, all we've done is we've swapped out the metal. That's it. It's the same construct, but we've swapped out the metal. Now lutetium-177, this is a beta minus emitter. So we can't use beta minus to image. If you put that on a PET scan, tried to image it, you wouldn't see anything. But those beta minus, because this has targeted to the tumor, the irradiation from the beta minus is going to attack the cancer cells. So then you get an image like this after a follow-up from the treatment, and none of those lesions are there. And this was FDA approved in 2017 as Lutathera. So there's a lot of that work continuing to ongo at Berkeley Lab. We are doing a lot of different things trying to swap out, and like you said, that's exactly what the research is. It's what cancer are we trying to target, what is our targeting vector, and what kind of chelator do we use? How can we make sure the biggest question, one of the biggest problems, right, is we don't want that radioactivity falling out of what we've administered. We want to make sure that that whole thing stays together and doesn't go anywhere except for to the tumor. Because there's also problems if you try to inject it into someone and it goes somewhere else. Yeah.
because is the treatment that's actually happening, is it killing the cells?
Yes.
Okay. So
Yes.
Isn't that that's the main issue with cancer, is that like it's hard to distinguish the cells, but we need to kill them to get rid of it, but we don't want to kill all our cells.
Exactly, because a lot of the main, if anyone has known anyone who has gone to get a PET scan, it's similar to this. It's fluorine 18 is the positron emitter, and it's labeled onto a sugar molecule, but all it is just sugar. So sugar, if you imagine, it goes everywhere in your body, but when you have cancer, it's upregulated at those tumor sites. So, but that's not specific. So that's where this research is moving. It's how can we tailor this more specifically to the type of cancer you have. So to contextualize just a little bit of the research that I do at LBL, and I feel very privileged to be able to work with a lot of different types, different types of radionuclides. So more on the radiotheranostics side, you want something with a shorter half-life, right? I don't think I would be like, hey, put something cool in me, but most people are not going to volunteer for a very long half-life radionuclide being administered into them. So even lutetium Lutathera that has a 6.6 half-life, day half-life, right? And that's the time where it takes for approximately half the material to decay. So that doesn't mean it's all gone in 6 days. It means that lots of it is still there. So all the way up to uranium-233, which has a very, very, very long half-life. And that leads into a little bit more of decorporation work, and I'm just going to touch on that briefly. So we work with an animal model, which is a mouse model, and in order to do this type of work, we basically take our mice and we give them a tumor, but we have to give them a tumor that's in a very specific spot and a very specific size, so that we know exactly where it is, and so that we know if the tumor treatment is working. So we will literally, for tumor treatment studies, we will give them a tumor, wait for the tumor to grow, and then we will give them the treatment, and we will take a little ruler, little calipers, and just measure, hopefully, the size of the tumor shrinking. But that's basically how we do those mouse models, and very conventional, actually, mouse model. There's another type of cancer, not cancer work, but work that we can do using this idea of chelation and radioactivity. And that's to kind of the other way around. What if you've accidentally been exposed to radioactivity? Or what if we, for example, needed some kind of treatment if we were exposed to radioactivity? So that is kind of a study called decorporation. The idea is, we get contaminated with something, how can we treat? So just really briefly to define this, because this is a lot of work that's funded by the Department of Energy and the Department of Defense, that we get a lot of funding for at LBL, and I can't disclose all of the research and results that we have, but I will tell you about a little bit of what we do. So radiocontaminants, basically radioactive particles or unstable elements released into the environment. We've had lots of examples of this before, some of it pretty recent and relevant, right? In the case of a nuclear disaster, you will be exposed, then you will have uptake of that radionuclide, and it will distribute and accumulate. If you're exposed to a radionuclide on its own without being preformed into something safe, it will go to your bones. You will excrete it slowly, but it will go to your bone, and that's just what happens. So decorporation is the idea that we're going to take some of this contamination healthfully and release it from the body. So rather than going to the bone, it's going to distribute and accumulate and then be excreted. Apparently I missed that animation here. So a lot of the work that I do at LBL is using this thing, which maybe doesn't look like a chelator to you, but it really is. It just grabs onto the radioactive metals like that. We call it HOPO. It just has a really high affinity for metals, so it will rapidly capture it and transchelate it. So I guess I took that slide out. But basically, when we do our decorporation studies, we, in a very controlled way, contaminate the mice, and then we wait a certain period of time, and then we give them the treatment, and we basically monitor, is the radioactivity being cleared from the body? So just summary of topics today. So hopefully what I would hope you take away from this is that at LBL we're doing a lot of really cool stuff. There's theranostics, which is cancer diagnosis and therapy, and it's the idea of using chemistry and chemical design and biological design to piece together a compound that can both image and treat cancer. And I showed you one example of an FDA-approved drug. There are actually several more FDA-approved theranostic agents, and much more is coming out. It's a very hot field right now. Also the idea of decorporation and the fact that we can do both forms of this type of research at LBL at Berkeley Lab, affiliated with UC Berkeley, is what makes it just really unique and really amazing. All of the different isotopes that we have access to. So with that, there's a bunch of resources, and I'm happy to answer any questions regarding any of this. You can really just look up the United States Environmental Protection Agency has a lot of information on radiation protection. Some of the slides on, or some of the images on shielding I literally got from the Oak Ridge Institute for Science and Education. Oak Ridge is a large producer of radionuclides, so they have a lot of information. I also encourage everyone to check out clinicaltrials.gov. That has where all of the current theranostic work undergoing. And then my boss, Rebecca, she actually has a company based on her, that chelator, HOPO, that is actively going into human trial, or is in human clinical trial. So just to plug that as well. So with that, I would like to thank my huge team of people that helped me do all of this work. It's really because of them that all of this gets done. Animal studies, you probably know, take a huge team of people and a lot of training and patience. So yeah, and thank you again to the organizers. Yeah.
Do we have any questions?
Which stage of cancer actually this one is targeted at? I guess in a lot of cases it's most effective just kind of doing operation, get a tumor out.
Mm-hmm.
And what kind of cancer is best to use this type of treatment?
This is a general kind of, we're trying to, more in a general sense, trying to create these new molecules and new drugs, but what we're really targeting with this type of stuff is really targeting are the very, very small metastatic cancers, the ones that are just extremely difficult to try and conventionally image or treat. So tumors that are very have a lot of heterogeneity, have a lot of different things going on. That's what we're really trying to target, the tricky stuff.
And this will still kind of harm the host healthy cells too, right?
No, they don't. Yeah, the goal, and that's a lot of what we look at, is making sure that these molecules that we're designing are only going to the cancer cells and not the healthy cells.
Okay. Is the radiation, once it's grabbed onto the cancer cells,
Mm-hmm.
but radiation is all directional, right? And
it will stay. The radiation will stay at the cancer site because of that targeted interaction. But that's actually something that a lot of people are looking into, is how much of the radiation stays and how much gets redistributed. Okay. Yeah, it's an important question.
So is that the different kinds of particles that they emit and the penetration of them?
Yes, and that's the, not every radioactive isotope that's unstable just perfectly decays to one daughter. A lot of these isotopes have complicated decay chains, and they decay to all kinds of crazy things, and you have to think about where's everything going and what's the half-life of everything in the decay chain. It gets kind of complicated. But yeah.
Yeah, it's a real basic follow-up. Is the idea that the lowest half-life is safer, or that there's a middle zone that you prefer, or just that it has to match your targeting dynamics? Okay.
That's exactly it. Yeah. It really depends. For example, antibodies have a really long circulation time in the body. They just take a while to reach the tumor site. So we use a breast cancer model for a lot of our work, and that a very common targeting vector for breast cancer is antibodies. So when we go to do breast cancer research, we have to pick a slightly longer lived radionuclide. So that's, for example, why zirconium-89 and lutetium Zirconium-89 is one of the most popular in the field of imaging because it has a 78-hour half-life, and so it gives us a few days to image. Yeah, it's a good question.
What was, or I forget what the term was for when all of the particles are in the body, or like when we inhale
The decorporation.
Yeah.
Yeah.
So that causes cancer because you have all of the decaying particles in the body, and they will radiate it, and like when it hits the DNA in the nucleus.
Yes.
So is that a different process than like if you're exposed to radiation externally, like from some sort of, I don't know.
It's
like how
the
else would
same.
you be? Well, how else would you be exposed to it? And it's just the same thing versus like a physical metal in your body versus whatever particles hitting you.
Yeah, it's the radiation is radiation. So whether it's being your skin outside externally is being radiated, or whether you've inhaled it. But then you also need to think about what type of radiation, because alpha radiation is shielded by a piece of paper. But so, yeah. Yeah.
Great talk.
Thank you.
This might also be like a very simple question, but could like the treatment versus like the imaging, radionuclide that you use have different like off-target binding when it's administered?
Yes, because it's actually one of the most difficult challenges in the field is matching your imaging radionuclide with your therapy radionuclide, because they're different they're different molecules. So one could be bigger, one could be smaller, one could prefer a more acidic ligand, like a ligand that's gonna hold on to it tighter. One might prefer something that's larger, and so it's actually a huge issue and a big thing of debate. I showed you that they used gallium-68 and then lutetium-77 for the match of the imaging and then therapy, but those two metals are actually pretty different. So one behaves a little bit more different in the body than the other, and that's a huge problem. So yeah, it's definitely something that we have to think about when we're designing these. Yeah.
If you're designing a treatment that's based off of proteins, would you also need to administer other drugs to change the protein response in the body?
Not necessarily, but sometimes. Yeah, it kind of depends. But yeah, it's a good question. Yeah.
How effective are these treatments, and how popular or widespread are they? It sounds like something, this is like personalized medicine or cancer treatments are new.
Mm-hmm.
Yeah, just curious.
They are very widespread. I wish I'd done a better job at giving some more examples. Some over the past, 10 to 15 years have become FDA approved by the Food and Drug Administration. So the radioactive metals attached to a ligand with a chelator and a targeting vector are not necessarily new. But what I've shown you today is more on the cutting edge side of where this research is going, is kind of what I was trying to give you more of a flavor of. But yeah, it's not as, it's up and coming in general. In the past 15 years, it's really been developed, so.
And the development is personalized medicine versus more general treatment?
Yeah, the development specifically on a more chemistry fundamental side is about creating that personalized medicine approach, right? Versus it's doing away from one drug to do it all. Well, it's one treatment, it's one thing. It's no, everyone's a little different, and every cancer is a little different. So, yeah.
Okay, well, if that's what we have, thank you so much, Alexia.
Thank